Dear students, we have completed the introduction to physiology's first lesson in the classroom. As we were not able to do the second lesson in the classroom face-to-face, I have prepared this video as your second lesson. So please send your questions, if you have any, to me via bitajoglu@ciu.edu.tr. I will write this in the explanation part of the video. You will be able to see my email address. Now we have to carry on with the cell and function and cell and organelles and structure of the cell membrane, because these will be the basics for understanding how cells function and how we are kept alive. The view that best explains the order in which the molecules are arranged is the fluid mosaic membrane model. According to this flowing mosaic model, the main roof of the membrane is a double layer of fat. Fats are also termed as lipids. Large and small protein molecules are irregularly embedded in the fat layer, which gives it a mosaic appearance. Carbohydrates bind to some of the proteins and form glycoproteins. So when I say glycoprotein, you will know that it's a carbohydrate which is bound to a protein. And by binding to fat molecules, they form the glycolipids. So when I use the term glycolipids, you will know that we have carbohydrates that are bound to the lipids. These molecules play a very important role in the selective permeability of the membrane. By these, cells can recognize each other and take special substances, such as hormones, into the cells. As you can see here, the phospholipid bilayer forms the cell membrane, with the phosphates facing the extracellular fluid, and from the inside the intracellular fluid, with the hydrophobic ends, which are formed from the lipids facing each other, and hydrophilic heads facing the liquid parts of the cell, whether it's the intracellular or the cytoplasm of the cell, or it is the extracellular fluid. And we have many features attached to the cell membrane which do specific jobs in order the transportation of the material that has to be taken into the cell or has to be excreted out from the cell. Therefore, the structure of different species may differ from each other. One of the most important features of this model is that the lipid layer is constantly moving and flowing. The pores in the cell membrane are the responsible structures for the selective permeability of the membrane. So the structure that you see here has specific pores, contains specific pores that are responsible for taking specific structures into the cell and excreting them. The size of the materials that are going to pass through the membrane is decided by these pores. The majority of the lipid layer of the cell membrane is composed of these special molecules, namely phospholipid molecules. The phosphate arm of the molecule has an affinity to water and binds water. The fatty arm is aquaphobic, which means it doesn't like water much and repels water. So therefore, the circles that you see here represent the phosphate arms of the structures that form the cell membrane, and these long strings represent the lipids, the fatty arms of the membrane. So the fatty arms face each other. They do not get in contact with water, and the phosphate arms are in contact with the cytoplasm within the cell and with extracellular fluid outside of the cell. As the structure of the cell membrane is developed, aquaphilic phosphate arms direct themselves towards water, and aquaphobic hydrocarbon chains repel themselves away from water. Aquaphilic means loving water, liking water, and aquaphobic, as I said, doesn't like water. As a result, the phosphate arms that have an affinity to water, both in the inside layer and the outside layer of the membrane, position themselves to face water in and outside the cell. The sequencing of the membrane during development is extremely important. It is the phosphate arms that face towards outside and inside within the cell membrane enables the cell to be able to take water inside, and this is imperative for a cell to survive. And of course, with water, there will be other solute materials that will be entering this intracellular space into the cytoplasm of the cell, which are very essential for the lifespan, for the life performances of the cell, living performances of the cell. In other words, phospholipids are attached by the tails and form a double-layered membrane, as can be seen in the picture here, which is an enlargement of this structure here, and you can see the hydrophilic head is constituted of the phosphates, and the hydrophobic tail is constituted of the lipids, phospholipids. So they face each other with the hydrophobic arms to form a double layer cell membrane. Aquaphilic heads faces the water-based cytoplasm in the cell, and the water-based intracellular, or my apologies, or the water-based intercellular or extracellular fluid outside. So if we consider this part to be the inside of the cell and this part to be the intercellular space or extracellular fluid, the fluid which takes part within between the cells, the both hydrophilic ends of the cell membrane meet with the watery sides, with the hydrophobic arms being facing each other at the inside of the layer, the membrane. Those who are caught between the aquaphilic inner and outer surfaces of the cell membrane are water repellent tails. These are the tails. So fluid mosaic model is explaining us the double layer of the cell membrane with many other specific areas on the cell membrane, like the globular proteins, glycoproteins, carbohydrates attached to them, glycolipids, cholesterol. As you can see, cholesterol is always present in the cell membrane, and peripheral proteins and protein channels, which also enable the transport of certain materials that we will be talking about in the coming future. When we look at the functions, that was the structure of the cell membrane, and when we look at the functions of the cell membrane, it is a thin membrane that forms the outermost layer of the cell. It separates the intracellular cytoplasm from the extracellular environment and other cells. The cell membrane determines the boundaries of the cell and ensures its integrity. The complex structure has a selectively permeable layer which regulates material input and output to the cell. The glycoproteins and the glycolipids found in the cell membrane give antigenic properties to the cells. Now, if you haven't heard anything about antigenicity, I'm sure you did, but if you haven't heard about what is antigenicity, is this. Every species, or even human beings among themselves, have specific protein sequencing for specific tissues. And when these sequencing in the tissues, protein sequencing in the tissues, is not completely the same with other people's or other species tissues, then this will act as an antigen to the host, to the receiver, and the receiving body that received the tissue will develop an immunogenic reaction towards these sequenced proteins, which is called the antigen. So the glycoproteins and glycolipids found on the cell membrane give antigenic properties to the cells. This is to give each separate cell a different feature or an identity. Some proteins act as enzymes to speed up chemical reactions. So when we say enzymes, we know that they're in a protein organization. They're in fact proteins, but they're specific proteins to catalyze some activities, chemical activities, and increase the speed of these chemical reactions. And they're called enzymes. A cell can communicate with other cells and substances in the intracellular fluid via its receptors on its surface. Now let us take a look at the transport through the cell membrane. One way of transportation through the cell membrane is by diffusion. Diffusion is the movement of a substance from a higher concentration to a lower concentration. So the shift of the diffusing material is from a higher concentration area to a lower concentration area. For example, think about a scent that spreads through the room, or a drop of ink stains the whole glass when put in a glass of water. it continuously becomes diluted. The same rule applies to the cell. For example, glucose is constantly consumed in the cytoplasm inside the cell, and the density of residual substances increases. The difference between these densities, for example, what I mean here is that when glucose is constantly used within the cell, its concentration will get lower and lower, but glucose at the intercellular space or extracellular space is higher than the concentration of glucose in the cytoplasm of the cell, because it's continuously being consumed in the cell. Therefore, this concentration gradient leads to the diffusion of glucose from the extracellular fluid towards the inside of the cell, intracellular fluid, which is the cytoplasm itself. So the substance to be transported by diffusion through the pores must be small enough to pass through the pores and must be water soluble. It must be able to dissolve in water because they pass with water through the pores of the cell membrane. Large molecules cannot pass through the pores, and these molecules have to be intaken into the cytoplasm with different diffusion methods or taken methods. For example, while glucose is transported by diffusion, starch, which is a bigger molecule of sugars, cannot be transported with this method because it's a quite big molecule. So higher number of pores increases diffusion rate, and there is no need to use a size limit or pores to transport fat-soluble substances by diffusion. Since the cell membrane is lipid structure, these substances can pass through any part of the membrane, like oxygen, like carbon dioxide. The other way of diffusion is simple diffusion. The transportation through the cell membrane is first by diffusion and the second by simple diffusion. Fat-soluble substances diffuse through the double layer of lipid layer. Water-soluble substances diffuse through the water-filled channels of carrier proteins. The most important factor determining the diffusion rate of fat-soluble substances is its solubility in fat. So solubility of oxygen, carbon dioxide, and alcohol is high, so they can diffuse as if there is no membrane present. So highly soluble solutes can what I mean highly soluble solutes in fat, because there is a lipid area, as if you are to remember the double layer which had lipid layers facing each other at the membrane. Since they're fat soluble, these can pass into the cells as they're dissolved in fat, as if there is no membrane at all. And oxygen, carbon dioxide, and alcohol are examples to this. Water, on the other hand, passes through the protein channels even though it does not dissolve in lipids. So different mechanisms take place for diffusion of different materials into the cell and out of the cell. So as the molecular size increases, the rate of passage decreases because there are certain channels that are capable of transporting things inside the membrane from the extracellular space towards the intracellular space. So this is outside the cell, this is inside the cell. As fat solubles pass through the membrane, some water solubles need to attach to the protein channels to pass through the cell membrane into the cell. Protein channels open and close and are selectively permeable. Heat increased diffusion area and decreased membrane thickness increase the diffusion rate. So the sodium and the potassium channels are very important in a cell's life, especially the muscle cells that are to contract and cardiac cells which need contraction continuously. Important, and these channels regulate the passage of these ions. So if we look at the principles of the mechanisms of the protein channels in simple diffusion, there is a voltage door and there is a chemical door. The voltage door is the gate that is sensitive to electrical voltage changes in the cell membrane. And sodium and potassium ions are transported in this way. So we need a voltage door there. The chemical door, it is opened and closed by the attachment of a molecule to the protein in the channel structure. For example, the acetylcholine of the autonomous nervous system, which we'll be seeing as we come to the nervous system physiology. The third type of diffusion from the cell membrane into the cell is called facilitated diffusion. The passage of water and fat-soluble substances as well as sugars such as glucose, galactose, and fructose and amino acids through the membrane occurs through a pathway called facilitated diffusion. The substance to be transported combines with the carrier protein in the membrane to form a new compound that dissolves in the lipid and is then transported into the cell. After the material transport is completed, the carrier protein takes its original shape again and separates from the ingested transported molecule. The transportation occurs from a high concentration medium to a lower concentration medium. The increase in the number of pores accelerates the facilitated diffusion. So pores are the most important areas in the cell membrane to take necessary elements into the cell. When the substance to be transported binds to the carrier protein, the carrier protein changes shape and the end of the protein channel at the stimulation site opens. So stimulus molecules come to the binding site, it opens the channel on the cell membrane, and the molecule that is to be taken into the cells can pass through this protein channel and goes into the cell to be utilized. The molecule starts to enter from here, the opened end of the outside of cell. This is the intercellular or space or the extracellular fluid, and this is the intracellular fluid. So when the molecule in the intercellular space comes to the end of the opening towards the intercellular space, within the stimulus molecules binding to the channels, it opens the channel and the protein passes in, or the any substance to passes in. Despite the facilitated diffusion seems to be similar to active transport, The major difference between them is that facilitated transport does not consume energy, and the movement is always from a higher concentration towards a lower concentration. Sorry for the spelling, it's lower there, but it's lower concentration. So we will be seeing active transport now, but the difference between the facilitated diffusion, the major difference is that facilitated diffusion does not consume any energy, and the transport is always from a higher concentration to a lower concentration media. The other way of transportation through the cell membrane is called osmosis, and I am sure that you are very familiar with this word, with this transport method from your high school knowledge. The density of a substance is the amount of substance in solvent per unit volume. In the case of high solubility, the medium is very dense, and in case of less solubility, it is less dense. The density of the medium is inversely proportional to the amount of solvent, and naturally, this difference in concentration needs to be balanced. So substances such as sugars, organic and inorganic salts, creates an osmotic value in the outside of the cells. Depending on the density of the cytoplasm and the external environment, which means the extracellular fluid, water passes between both environments through the cell membrane that acts as a semi-permeable membrane. Accordingly, the transition of water from an environment where it is more through a semi-permeable membrane to an environment where it is less is called osmosis. It is also possible to see this phenomenon in living things. In living things, the closed environment in the cytoplasm limited by the cell membrane. Osmosis is the transition of liquid molecules through the semi-permeable membrane from a less dense environment to a high dense environment to maintain the balance of their densities of the both sides. So if we have a semi-permeable membrane here and a dense solution with a lot of solute in the solution and a less dense fluid at the other side of the semi-permeable membrane with less solute and more water, water passes through the semi-permeable membrane due to the high concentration here and tries to equalize the concentration of both sides, and this is called the osmosis. The cell membrane creates a boundary between the intracellular fluid and the extracellular environment in the living things. So osmosis continuously happens. According to the density differences of these two media, water passes through the cell membrane, and this transition continues until the liquid concentration reaches to an equilibrium. When the cell is placed into a more hypertonic medium than it wants to balance the density on both sides of the membrane by giving water to the dense environment. So the water is lost, the water within the cell goes outside to a more dense area. That is what is meant with hypertonic. It is more dense than the medium of the cell inside. And as it gives water to the denser medium, it shrinks by losing its water. The shrinkage of the cell when placed in a denser environment, hypertonic environment, is called plasmolysis. But if the cell is placed in a less dense or hypotonic, not hypertonic, but hypotonic environment than itself, water enters the cell in order to bring both sides of the fluids to an equilibrium. Therefore, it swells by taking water inside. And at a certain swelling, where the cell membrane cannot stand the swelling anymore, it bursts out and the cell dies, which is called deplasmolysis. So the shrinkage of the cell by giving out water is called plasmolysis, and the opposite is named as deplasmolysis. Now let's take a look at what osmotic pressure means. Osmotic pressure is defined as the pressure of the liquids exerted on the cell membrane as they enter the cell due to the density of the substances within the cell. Substances that create osmotic pressure are various sugars, organic acids, organic and inorganic salts. Therefore, the density of these substances in the cell is directly proportional to the osmotic pressure of the cell. During deplasmolysis, when the cell is in a less dense medium, water enters into the cells and starts exerting a pressure from the inside onto the cell membrane. As the critical threshold is reached, the membrane cannot handle the pressure from the inside and loses its integrity and bursts out. Hence it dies. So, for example, if red blood cells are placed in a high density medium, water will enter the cells and swell and eventually burst them out. And when this happens with the red blood cells, the specific name of the disintegration of red blood cells cell membranes is called hemolysis. During deplasmolysis, when the cell is placed in a less dense medium, water enters into the cells and starts exerting a pressure from the inside onto the cell membrane. As the critical threshold is reached, the membrane cannot handle the pressure from the inside and loses its integrity and bursts out. For example, if red cells are placed in a low density medium, water will enter the cells and swell, and eventually the red cells' membranes will burst out. And this specific event, the bursting out of red cells' cell membranes, is called hemolysis, which means that the blood is not able to carry oxygen to your tissues anymore. So I would like to give a quick example for this osmosis and osmotic pressure in real life. When a person is drowned in seawater, because the seawater is salty and it's a more dense medium than the human body, and when during drowning the person swallows lots of salty water, the medium will become denser within the blood as salt is taken into the bloodstream. So the red cells in the bloodstream will give their intracellular fluids to the blood plasma, which is denser now due to the salt ingested, and they will shrink, which is plasmolysis. And if these persons are to be treated with proper dilution therapies, because the red cells are plasmolyzed and they shrink, they can come to an equilibrium as we give normal serum to the patients, water-based serum, to bring the plasma of the blood to a equal density with the red cells. Then the cells will take water from the bloodstream and come to the normal shapes and will be able to carry oxygen. However, if a person is drowned in a river with no salt solute in the water, then this water will be hypotonic. And when hypotonic river water is swallowed in enough amounts to make the plasma of the blood less dense than the red cells, then because the hypertonic medium is within the red cell cytoplasm, water from the plasma of blood will enter into the red cells, and eventually it will swell and burst out, which is deplasmolysis. And in the end, the bursting out of red cells will lead to hemolysis, and there will be no red cells around in order to be able to carry oxygen anymore. Therefore, drowning in a hypotonic medium is far more dangerous than drowning in a hypertonic medium. So this is a good example to explain the plasmolysis, deplasmolysis, with the osmotic pressures that fluids exert towards the cells in the body. So here is a very good example of the red cells. As you can see, in isotonic solutions, where all our bodies are composed of isotonic solutions normally, and it is kept like that with many reactions. The cell and the outside cell medium are isotonic. They are equal in density. So the cell neither swells nor shrinks. So this is the normal appearance of a normal red cell with a round body and with an indentation towards the center of the cell. And when you look under the microscope, this is what you see for a red cell. But when you put the cell into a hypotonic medium, then the cell will, because it is more, it has a more oncotic pressure towards the outside, because the outside is hypotonic, the water will enter into the cell and it will make it swell, and the one step forward, this cell will burst out. But if you place the cell into a hypertonic medium, then the cell will give its water outside and shrink and will look like something like this. And this is a very good example of plasmolysis and deplasmolysis. This is plasmolysis and this is deplasmolysis. So under normal conditions, the amount of water that travels bidirectionally in and out of the cell is so sensitively balanced that the volume of the cell remains constant. And this is explained with the word homeostasis, which I have talked about during the first lesson in the classroom. So when we say isotonic is osmotic pressures are equal inside and outside of the cell. When we say hypotonic, the osmotic pressure of the medium outside the cell is less. And when we say hypertonic, the osmotic pressure of the medium outside the cell is high.